Okay, now, so first of all, welcome everyone to the second online seminar from our ISDCI seminar series. For those who are not familiar with the society, it stands for International Society of Developmental and Comparative Immunology. And well, I'm Maria Verlens. I'm the Secretary of Education of the society. And basically, since this year, we started this online seminar to, of course, promote the society, but also to reach a little bit of a broader audience. And like in a presentation like the one of today, to see also whether we can bridge to other well-established and existing society, for example, the zebrafish society. Some of us are members of both societies, and we find that some topics are actually very useful for members of both of the societies. And this also brings me a little bit to the speaker of today, Julien, who is one of our young members, young and promising and rising stars of our society.
You will make me blush.
Really? I can't see it. He did his PhD in France, and then since I think 3 or 4 years, you're already in Norway, the country you always wanted to be, in Oslo, to be more precise. And now we know all Julien because he's also very well known also among the zebrafish society, because our zebrafish rock guy always promotes many of the beautiful pictures that Julien produces of his zebrafish, and not only zebrafish, mostly of the gills, but not only of the gills. So Julien is really a master on immunohistochemistry, as I said, on zebrafish or many other fish species. But then thanks to his love for immunohistochemistry and really looking and fiddling in the organs of the fish that he studies, then he also ends up discovering new organs, discovering new cellular organizations. And that's why today we invited him to tell us a little bit about his journey into the bronchial cavity of the fish. And this journey also awarded him a very nice prestigious grant, at least here in Europe, the ERC grant. So you were recently awarded the, I think, starting ERC grant. So congratulations also for that, Julien. And with this, I'd like to leave the screen all to you. The floor is yours. Take us into your journey.
Thank you a lot for the invitation, a very kind introduction. So yes, so right now we'll try to explore a bit the journey, like a special journey into the bronchial cavity of the fish on its immune system. Very quickly, for that I'm the University of Oslo in Norway. I started my group in January this year about spatial immunology and the use of nanoparticle. I'm part of the comparative immunology groups at the Department of Bioscience, which is right over there. And together with the groups of the Professor Shuang Kiao on Feneric Johansson's that works more in code, my research interests are more focused into the spatial dynamic of infections and immune responses, so especially in the bronchial cavity area, but also with the use of nanotechnology to try to study those immune mechanisms and to see if we can make new treatments. In the last years, I did also a lot of imaging and got a few rewards for my microscopy images by the Nikon Small Board competitions. I'm also involved in some outreach activity, for example, in the Howard Hughes Medical Institute, Janelia program, Beautiful Biology. My group is very young, I said, from January, and is composed at the moment of Martin Falk, which is a researcher, and Eda Hansen, master student. And in April next year, then there will be a new position opening for a postdoc. So why is the bronchial cavity important? So we can see it also linked to aquaculture, which is a crucial source of sustainable protein for humans. Half of the fish that we eat come from the aquaculture. Unfortunately, this industry is threatened by diseases, infectious diseases and injuries, which arise great concerns for food security, animal health and welfare, but also the public opinion, especially when you have pictures like this of very sick salmon that appears in the media. So new treatment and disease management strategies are needed to actually address this challenge, which requires a good understanding of fish immunology. And at the moment, immune responses in the field are mostly focused on analyzing the spleen and the kidney, which are the main systemic organs for immune responses. The study of the mucosal immune system is a bit less understood, and this especially applies to the bronchial cavity, which, if we think about it in the fish in the natural habitat, is exposed continuously to microorganisms, toxic agents, pollutant, and food particles. And in aquaculture, it's also involved in many diseases. It is thought to be a portal of entry for many infections. So you can see, for example, here, the gills of a happyfish, and how it looks when it's sick. So there is this issue that the defense mechanism of the bronchial cavity seems a bit overlooked. And if we try to see what is the bronchial cavity in terms of anatomy, so it forms two cavities in the head of the fish, one on each side, which are connected in the middle by the pharynx. They are lined by a mucosal epithelium. Each cavity contains 4 gill arches for gas exchange, and on top of each cavity, you have a thymus lobe. So these very important regions where we have vital organs, for very long has been perceived to have a very simple associated immune system, just made of the thymus lobes and scattered immune cells. And if we look a bit more into then the gills, we focus into the gills on how it looks. This is what you would see. So each gill has a more solid part, a supportive part made of cartilage and other tissues called the gill arch. This pointy part here sieve out food particles, they're called gill racers. Then from this gill arch, you would have filament, so small tree having little lamellae that you see here. Those are called filament. And it is on those lamellae that you see the gas exchange, or the gas exchange occurs. These filament in some fish in the middle are somewhat bound together by a septum. So we can see that a bit later that not all fish have a septum, and sometimes looks a bit different. If we look now, if we just make like this section across the lamellae, this is what you would see. So the inner part, so towards the septum, you have this vessel which is low in oxygen. The blood flow will then go, go from this way, from the low oxygens to the lamellae, and then the water flows countercurrent. And the oxygen will then get into the blood and leave the gills filament through this efferent artery. And this is to the right all the zebrafish gills look like in microscopy. If it's stained for the cell nuclei in blue and actin filament in orange. So you see the gill arch, the supportive structure. Here you have the pharynx. This little pointy part are the gill raker to sieve out the food. And there you see the filament and the little wings, which are the lamellae where the gas exchange occurs. So, as I said, for very long, all this has been perceived as having just a very simple immune system. But in 2008, actually, Erling Olaf Koppang showed that there was the first evidence of something a bit more complex. He found a new lymphoid tissue, he called the interbronchial lymphoid tissue, in the gill of Atlantic salmon. So this is how it would look like. So we have the gill arch, the supportive structures, and all the filament. If we make a section this way, this is how it would look. Still these supportive structures, one filament, the other one, and you have this septum here. This interbronchial lymphoid tissue that we will call ILT is lying right on top of the septum and rises a bit on the inner part of the filament. And now if we make pictures right on it with microscopy, so here we've followed into actin filament in green, cell nuclei in blue, and T cells and NK cells in magenta, this is how it looks. So you have like a proper aggregate of T cells, or organized aggregate of T cells. And Erling Koppang over the years could show that this interbronchial lymphoid tissue in salmon was not really a primary lymphoid organ, but their functions are like features of secondary lymphoid organs. And this is a bit at this stage that I joined the effort to characterize the gill immune system that was several years ago. I joined a study with Alf Dallon, Erling Koppang, Riyad Vergetiès, Pierre Boudinot, and Louis Dupasquier to study the presence or absence of this lymphoid tissue in the gill of different species. So we had specimens from like shark to trout, eels, zebrafish, and sturgeons. So we had all these samples to make section from. And then we use an antibody that was developed for fish, found by Maya and developed for labeling in fish by Maya Folensa, to label TNNK cells. So it's an anti-ZAP70 antibody. So ZAP70 is found in both those cell types, and it's highly cross-reactive. So it means it will react from human, guinea pig, shark, and a lot of other fish species. Not 100% of them, but most of them. And then we just did microscopy. So if we look in shark, in the gill of shark, so we add cat shark, you see here the supportive part, you have the filament, and you can see that the septum actually goes all the way and binds to the skin. So in shark, we don't see an ILT, but we see these thymus tissues inside. So in shark, we did not find any ILT. But if we go into teleost fish, on Chondrostean, which is like partially ossified fish, you can see that the septum is no longer continuous with the skin, but it's still like quite high up into the filament. And if we zoom there and make an anti-ZAP70 labeling, which will be in brown right here, you find an ILT in Siberian sturgeon. If we look then in more basal teleost, like salmonids, cyprinids, and eels, you can see that the septum, the side of it like shrink. It's just halfway or 40% halfway through the filament. But if you still make images right on top of it, you still find this ILT. And why it gets interesting is if we look into the gills of Perciformes, you can see that this continuous septum is lost. Because of that, the angle between the gill filament, opposite gill filament, is wider, likely promoting a bit more efficient oxygen uptake. But in this fish, then we don't see any ILT. So we tried to see if, so it seems that so far the presence of ILT follow taxonomic rules. And we tried to see if it was also affected by size variation. So for SIM, we know it was present in cyprinids or large cyprinids like European carp. So then we looked into zebrafish, which is another cyprinid, a lot smaller. And in this case, you have the gill right here on this section that you see is oriented this way. So here we are right on top of the septum. Phalloidin is in green, so actin filament in green, cell nuclei in blue, and T cell and NK cells in magenta. So here you have the septum. So here you are right on top of the septum. You see like opposite filament, and you can see this ILT there. So it was not impacted by size variation. So also you can see that, okay, what we see in carp, on salmon, we found it in zebrafish as well. It was quite interesting. We did the same with Medaka fish, which is a small beloniform, so small perciforms. And in this case, you can see that this septum actually is very small. Very quickly, they are separated, and then you don't see this ILT. So at this stage, we could say that in certain fish, the bronchial cavity contained 10 lymphoid structures, 2 thymus and 8 ILTs. And from there, I started to decide to use the zebrafish to further characterize the gills and the bronchial cavity, saying like, okay, it's a very small organisms, so I should be easier to characterize the organization of the bronchial cavity rather than if I take a big fish like carp and salmon. So very quickly, the zebrafish is a small freshwater cyprinid, raised between like 10 to 30 Celsius degree. It's easily accessible, short development, a lot of practicalities using this fish. It's also a well-characterized model. There's a wide community using this model to study human diseases, and we benefit from a lot of genetic tools. Close conservation to mammal, I would just go on and on, just heavily used. And to me, the main point of using the zebrafish is for the optic of it. It's easy to make live imaging, so you can observe live processes like as they are doing without any alterations from fixations or manipulating it. But the other one is that it's so small that in just one section, you can observe most of the fish and all the connections between the organs. You keep a very good context, the spatial context of the fish is well preserved. And just to give an example, this is what you would see if you have a fish, I show this way, like this, and you cut it like that through the eye. So here you see actin in copper to gray, gold, copper, gray, nuclei in blue-green. So here you have the eyes of the fish. You have the nostril, and you can see the connections with the olfactory nerves, the olfactory bulb, the rest of the brain, with the cranium right over there, the muscles, the vertebra, and the spinal cord. So in just one section, you have access to all the different connections between the tissues, and you can just investigate multiple organs at once. And this image just to show this is the actual resolution of each of this whole image. So we have very, very good resolution in a very large area. So we applied this to study the gill associated lymphoid tissue. And this is a paper we published in 2021. And we realized that the zebrafish gill, so gill associated lymphoid tissue, is quite compartmentalized. So if you take the zebrafish gill this way and you cut like a salami transversal section like this is what you would observe. You have the gill arch, this supportive structure. You have the septum, and then you have one filament on the opposite one. You have phalloidin, so actin in green. So what you see here, for example, are main arteries. Nuclei in blue, and T and NK cell in magenta. And what we can see is that we do have scattered cells in some of the region of the gills. So, for example, inside the gill arch, inside the septum, and within the filament. But if you look on top of the septum here, then you can see, of course, the ILT that we described in the previous paper. And if we were to section exactly this way, that would be this way on the gill, again, you see it perfectly zigzagging, and when you get out of the ILT, you see in the rest of the filament, the T cells are a lot more scattered. What was interesting when we did the study is that actually on the side of the gill arch, there and there, we could notice clusters of T cells that were not described. And if we zoom further, do a bit more imaging, so then you have one gill arch, the next gill arch. So here you have the water, the filament, you have some muscles and some cartilaginous structures. Then you see actually a lot of T cells clusterized all together. And so, okay, maybe just we were looking with the sections to see that, so we needed to do proper 3D imaging. So instead of making labeling of clear sections, we labeled the whole gill arch. And in this image, so you have the whole zebrafish gill arch, taken pictures from the side. In cyan, you have the actin. In magenta, you have nuclei, and in orange yellow, you have the T cell and K cell. So you see the gill wracker, that's about the food, the cartilaginous structures, and then this filament and the lamae for the gas exchange. On this cluster we found at the base of the filament on each side of the gill arch, this is what you see here zigzagging. It is a continuous structure. So we called this new lymphoid tissue the amphibranchial lymphoid tissue, ALT. So we have ILT for interbranchial lymphoid tissue on top of the septum, and ALT for amphibranchial lymphoid tissue on the side, each side of the gill arch. Of course, we did some characterization in zebrafish, and for the ILT, we could find some of the features shown in salmon. Actually, we also find in zebrafish there was a very good transpositions between the two models. So we showed that those lymphoid tissue were organized by a network of reticulated epithelial cells that you label using cytokeratin. So here in red. So they form like a meshwork that will organize all the T cells inside. And this is a very important feature to say that this is not a loose aggregate of immune cells, but something more structured. We tried to look a bit at the functions, so we could show that it was not, they were not primarily lymphoid organ. So we use a zebrafish line called RAG2DS red. So RAG2 is important for VDG recombination. So this is the process by which lymphocytes would acquire their repertoire. So this is present in the thymus for T cells and the head kidney for B cells. So this is important features for primary lymphoid organ. So here you see in the thymus, all the red cells are the cells expressing RAG2. But if we go into the ILT, on ALT, we don't see it. And for the ILT, actually, these data are corroborated what was found in salmon as well. We could show the presence of MHC2 expressing cells. So here, using MHC2 GFP line. So MHC2 GFP is in cyan. So we see the MHC2 expression in the epithelial cell, which is described in many papers in fish now. And you have all these immune cells inside expressing MHC2. We're not going to far into the characteristics, just like we have several other features, like features of secondary lymphoid organs, such as T-cell proliferation. So here you have a PCNA labeling in magenta. So PCNA is expressed by all cells undergoing cell division. That actually match the ZAP70. So we have double positive cells in the ILT and the ALT. So it's not a primary lymphoid organ, and we have some features of secondary lymphoid organs. So the ILT, what we found in ILT in zebrafish, match a bit what was described in the ILT of salmon, and the ALT seems to basically have the same feature or similar feature than the ILT. Now we try to wonder, okay, we know that for the interbranchial lymphoid tissue, we see them mostly in chondrostean and basal teleosteans. What about this new lymphoid tissue, this amphibronchial lymphoid tissue? And this is why it is interesting, because so far the amphibronchial lymphoid tissue has been found in all the fish we analyzed. So we have European carp here with the ZAP70 in brown. We see it very well in salmon, which are also basal teleosteans, but we also find it in modern, like this percomorph, Atlantic mackerel, northern red snappers, Pacific barracuda, and here in red in herp medaka fish. So from that point, actually, if we just make a slight resume at that point, we have per gill arch, we have one interbranchial lymphoid tissue that you can see in yellow. And this is like if you observe from above, you would see it on top of the septums and going along the inner side of each filament. So that would be for the basal teleostean. And on each side, you can see here in green, you have the amphibronchial lymphoid tissues. So each gill arch has 3 lymphoid tissues. And if we know we came from, like we just have 2 times lobe to 10 lymphoid structures to actually 26, 2 times lobe, 8 ILTs and 16 ALTs. Then we opened up or extended our investigations to the whole bronchial cavity, not just the gills. And there is more. We focus into this region of the fish. So the lower part of the bronchial cavity that is close to the pharynx. So this is how it would look like in a section. So this is exactly how it is oriented. If I show quickly with this fish, it would be sectioned this way. We are below the pharynx. So this is the isthmus, so a bit of flesh that separates the 2 bronchial cavity below the pharynx. You have the bronchial cavity to the right, bronchial cavity to the left. You have the head of the uroial bone. Right here, you have the first gill arch, the second gill arch. You see the filament and the lamellae, same first gill arch, second gill arch. And here you have the convergence of the gill arch to this piece of tissues beneath the pharynx, separating the 2 bronchial cavity. Now, I will reveal this is the ZAP70, what we observed. We have this massive structure that is exactly at the convergence of the 8 gill arches in contact with the water. So right here. Like a very massive structure that is constitutive. We find it in all fish. It has a lot of T cells, and we called it the mnemosyne lymphoid organ. Just go a bit after on the characterization, but important things in those images, we could see that here you have the ILT of 2 gill arches. You have another one here. See that you can see that the ILT and NILO, so mnemosyne lymphoid organ, were interconnected. And if we looked at other sections, so in this case, we are sectioned this way. You have the first gill arch, another gill arch here. You would have the 2 ILTs here, 2 subsequent one. And this region here is NILO. So we have this constitutive structure bearing a lot of T cells that binds the 8 ILTs and 16 ILTs all together at the convergence of the 8 gill arches below the pharynx. But as you can see just from those images, is that it is quite difficult to picture what would be the 3D structures of NILO just from 2D section. So instead of going with section this way, we had a collaborations with Meyeva and Guinchi in Montpellier to do serial confocal tomography. So instead of labeling sections, we were labeling whole head of zebrafish with anti-ZAP70 antibody, phalloidine, and DAPI. We then embedded those heads in agarose, and then we had a robot that was taking the block to put it under a confocal microscope that will acquire one layer. the robot will take the samples, put it into a vibratome, the layer that was acquired will be removed, then put it back into the confocal microscope, acquire another layer, put it back to the vibratome, and so on. So I think for a whole weekend, we obtained around 700 sections through the bronchial cavity, and then using the ZAP70 and Palladin and DAPI, we could reconstruct the actual 3D structures of NILO. And this is how it actually looked like. So you would have the anterior part of the fish here, the posterior part of the bronchial cavity right here. So this is the part that is around to the urea bottom. This is how it looks. It's a very complex structure that looks like a dragon with antler. This part here that forms this antler, actually the regions where the NILO actually merge with the gill lymphoid tissue. And if we reconstruct, there. No, NILO will be in magenta, and then I reconstruct the ventral end of the gill in green. In cyan, you have the amphibronchial lymphoid tissue, and in blue, you have the thymus of the fish. And this is how it looks. Yeah. And then you can see, like, you can understand why it has this very specific shape. It's really because it is at the convergence of the gill arches and connect with the gill lymphoid tissue. And quickly, you can see that it is also quite prominent structures into the bronchial cavity. If we go to the next video, you can see in this case, I removed in green the ventral end of the gill arches, but you can see the overall head of the fish in green. So the anterior part is still here. You will see the eye of the fish. Then you can actually appreciate the localization of NILO within the head of the fish. So it has a very prominent localization exactly in between the two bronchial cavities, and actually make a link around the ureal bone, connecting the two bronchial cavity. So thanks to these 3D reconstructions, we could estimate the volumes, and using stereology, we could estimate the volume of NILO that is occupied by ZAP70 plus cells, so T cells and NK cells. So it's around a bit less than 50% of the volume, while it's around like 2-3% of the spleen volume that is occupied by T and NK cells. And with using also 3D reconstructions, we could estimate the volume of fixed T cells at around 66 cubic micrometer. And with that, we could estimate the amount of T and NK cells in this NILO at around 165,000 T and NK cells, a lot more than what we estimated for the spleen, which indicate that NILO is likely a very important structure of the fish immune system. Of course, we tried to characterize it a bit more. So importantly, to show that it was a proper organ, we had to show that it was actually, or verify that it was organized by this network of reticulated epithelial cells. So we have redone this anti-cytokeratin labeling. And in magenta here, you see all these cells forming this network. In the 3D reconstruction to the right here, you can see that it makes this little pocket of T cells in NILO. And this is a really typical feature of lymphoid organ to have this type of cells. Like in human, actually, we could show that these cells express also MHC2. So here you have the reticulated epithelial cells in magenta, and if we remove, you can see that they're still slightly greenish, so they express a bit of MHC2. So this is also like similar to what we've seen with the ILT and ALT. Right? So far, it's the same thing. We went then into electron microscopy. So Jens Wollmann, that is a very gifted electron microscopist in the department, made what he called a dynamic infrastructure map. So it did like more than 1,400 images that it just recomposed as if you were on Google Map. So actually, this image you see to the right from a 9-week post-fertilization zebrafish, you see like the section back right this way. So you have one bronchial cavity, the other one, the head of the ureal bone, and you have NILO right here. It works like Google Map. The more you zoom, then the resolution adapts to your zoom. So, yep, all the way. There you can see, actually, you could zoom in and then you could see like all the detail and all the junctions between the cells. So of course, we see all the reticulated epithelial cells. we can actually see that there is a basement membrane that separates Nilo from the surrounding connective tissue, so very important features to say that it was an organ. It is quite in contact with the water, and in some places, like this one here, you have less than 1 micrometer separating immune cells from the external water on everything you would have inside. And we did not see any vascularizations, but we could see a lot of endothelial vessels bordering the basement membrane. And in some cases, even cells crossing the basement membrane. So good. So we know that we have a proper lymphoid organ, but okay, it would be still interesting to know what is the cell composition. And we know we have around 55% of T cells that occupy around 50% of the volume. Neutrophils, dendritic cells, and macrophages would represent a bit less than 5%. So here we use different transgenic lines. So for neutrophils, we add this MPX GFP line in magenta. We add some MHC2 GFP. For dendritic cells, we use an MHC2 GFP background here in green with peanut agglutinin lectin. So here you can see one of the dendritic cells. And then we use this MFAP4 M-cherry fat acylated line from Meeva in Montpellier. Very good line for macrophages. And you can see actually all the different macrophages. They're just like very big cells. Actually, they just still account for just like 3% of the cells in Nilo. And for B cells, we used an anti-Bruton tyrosine kinase antibody that label B cells and plasma cells, but also macrophages in zebrafish. So we did this labeling on the MFAP4 M-cherry fat acylated background to distinguish, like to not count the macrophage that would be positive. On here you can see B cells and a few plasma cells. And those actually account for like 15%. So more than 70% of the whole population of Nilo is made of lymphocytes. And then we have antigen-presenting cells, and I guess that the 25% remaining are mostly the reticular epithelial cells and the mucosal epithelial cells separating the organ from the water. So it's really much like a lymphoid organ, but still we don't know much more about the functions. So we try ask for the ILT and ALT to see if it was a primary lymphoid organ. So one way is to check for hematopoiesis. So for this, you can use the CD4T1 GFP line in zebrafish, for which we have like thrombocytes. So here we are in the kidney of the fish with actin in green, DAPI in blue, and the transgene in magenta. Those very bright cells would be thrombocytes. But the low fluorescent one, as you can see here, are actually hematopoietic stem cells. So this is one way to see for hematopoiesis, and we don't find this cells in Nilo. So there's no hematopoiesis. And of course, we check also for this VGG recombination. So, sorry, I'm just losing my earpod. We could check for VGG recombination. You can see again in the thymus, a lot of expression of RAC2. You can see it in B cells in the kidneys, but in Nilo, you mostly don't see it. So it's not like it has no hematopoiesis, or this is not involved in VGG recombination. So it's not a primary lymphoid organ. And again, like the ILT and ALT, it does display features of secondary lymphoid organs. We see T cell proliferations using the same methods with double labeling with anti-PCNA antibodies and anti-ZAP70 antibodies. We see the presence of activated T and NK cells that would be ZAP70 positive and MHC2 positive. You can see with the cyan arrows. Cells, T cells that would express effector molecules, such as TNF alpha, such as these cells here. So it has different features. We don't know yet if it's a conventional secondary lymphoid organ or not. What we know is that it shares a lot of features of secondary lymphoid organ, and also a lot of features with ILT and ALT. Actually, they share like many similarities. And more strikingly, so even their network of reticulated epithelial cells is actually shared. So here you have those cells, this network in Nilo that connect with the ILT. So that would be actually important question to see, like, are the gill lymphoid tissue on Nilo just like two parts of one single structure or not? Where it got even more interesting is then if we look at all those three tissue react to infections, then we start to see differences. So with collaborators in INRAE, Pierre Boudinot and Dimitri Rigaudot, they infected zebrafish with spring viremia of carp virus by emissions, and we look 3 days and 10 days after infection. So if you remember, Nilo had a very homogeneous organization of T cells. Well, in this case, at 3 days with this virus, instead of these very smooth organizations, you see like a very striking clusterization of all the T cells. So we have like a very quick, just in 3 days, reorganization of the tissues and of the reorganization of the T cells themselves. We would also know, like what we showed in the paper, is that bordering these clusters, we would have like cells containing viral particles. So we don't know if those could be reminiscent of some of the reactions we see in mammals when there's immune responses. It's something we have to dig out a bit more now. But what was interesting is if we look a week later, these clusterizations start to smoothen up. You can see that it's a bit less pronounced and you start to re-get some homogeneity. So Nilo persists and is being reorganized. And interestingly, if we then look at the gill lymphoid tissue, the ILT is very different. There the ILT shrinks and you have like a strong depletion of T cells. We barely see any T cells in the ILT anymore. And in this case, if you look 1 week after, then it replenishes. And the same applies for the amphibian lymphoid tissue. So here you have the gill arch, so you're on the side. You see very little T cells. But they come back, like a week later, we see a bit more. So it seems that we have the first difference between this gill lymphoid tissue and Nilo. Nilo persists during these infections, while the gill lymphoid tissue depletes. I indicate that those two regions may have different functions or roles in the immune response or defense mechanisms to the fish to infections. And it is even more striking if we look with another virus. So the infectious hematopoietic necrosis virus, IHNV, same protocol. Here you see at 3 days, you start to lose a bit this homogeneous distribution. You have a slight clusterization that just go back to normal a week later. So it's a lot milder. But if you look in the gill lymphoid tissue, you still see this very strong depletion of the ILT. And a week later, you see that everything replenish. So we don't know if those cells die, or this is newly generated, or if it's just like migrate and come back. Something we also have to see. Then we had fish from Stephanelos in Singapore that were naturally co-infected with different pathogens, Pseudoloma neurophilia, Pseudocapillaria tomentosa in the gut, and Myxidium stress injury in the urinary tract. So Pseudoloma neurophilia is mostly in the nervous tissues. So 3 parasites that do not infect the bronchial cavity. And very interestingly, Nilo still reacts. You lose a bit this homogeneity, you have actually reductions of the amount or the fraction of T cells in Nilo. You would have like this small clusterization from time to time. So you see that the structure, like they still had adaptations, like infection somewhere in the body impact Nilo and also impact the gill lymphoid tissue. So whereas the virus make the gill lymphoid tissue depletes, parasite infection seems to actually make them grow bigger. So I think studying all those 3 tissues seems very important to understand how the fish react to the different type of pathogens. So I think so we'll dig more a bit in the future. So that was a bit for the function, but in addition to Nilo, ILT, and ALT, there was still something a bit more. Actually, if you go back to this image, when you had Nilo right here, the ILT, connection with ILT, the gill arches, you remember that the bronchial cavity is lined by a mucosal epithelium that just like line all the whole thing. Actually, this epithelium is also very rich in T cells, so much that it actually forms a lymphoid network that actually interconnect some kind of this cohesive unit made by Nilo, ILT, and ALT with the rest of the bronchial cavity. So right now, we made a section this way through the fish, right here, and we focus in just one gill arch. So you have the anterior of the fish, the posterior part, actually a bit of the heart. You have the first gill arch, the second one, third, and fourth. You see the ILTs, good. You see some of the septum, and you can see all there here, bearing a lot of T cells. This is this mucosal epithelium lining the bronchial cavity, and here you have Nilo. So it really forms like you have this wider lymphoid network that interconnects all of it. And if we then make a section this way, when you have the thymus, so you have cortex and medulla. So ZAP70 is less pronounced in the cortex, but LCK would, for example, be more expressed in this region. And then you can see like this lymphoid network, like interconnecting the thymus with the gill lymphoid tissue. So in a way, you have this kind of lymphoid superstructure that integrate all the 27 lymphoid structures of the bronchial cavity to just like one super unit, which is very interesting to understand, like, why would you have the thymus interconnected with structures bearing secondary lymphoid features. So like, this is zebrafish. We try to look then in other fish species because maybe it's just a peculiarity of the zebrafish. So we look in another cyprinids, the crucian carp that we obtained from Chani Lefebvre at the University of Oslo. So here the section is done this way. We are below the pharynx, so this is this pharyngeal isthmus. You have the head of the urogeal bone, one bronchial cavity. The next one, one gill arches, second, third, first gill arches, second and third. With actin in green, so you see very nicely the muscle fibers. Actin in blue and in magenta, those are T and NK cells. So here you have Nilo that is very prominent, actually, even like those black lines you see here. In this fish, Nilo is vascularized, or there is endothelial vessel inside. And you see the same connections with the ALT and with the ILT. So everything that we've seen in zebrafish match with the crucian carp. But you actually see the same thing in salmon. So here you have like one gill arch, the second one. So you have like one ILT, second ILT, and again, you have Nilo in between. On this black spot that you see here are melanoma macrophage centers. So we have to see if those ones have a similar function as the one described in the spleen in chart. So, but this is still like basal teleost. What about plecomorphs? Looking at very old paper, we could actually detect in 3-pan stickleback. So if you look for paper that study the pheroid, because the pheroid is below the pharynx in this isthmus, when this is where you would find pheroid follicles. So in the images made for those studies, we could actually recognize Nilo. So here you have the subpharyngeal isthmus. So in this paper from, I don't know, actually this one's a bit more recent, but in sometimes old paper we will find it. And then here, what you see there is Nilo. You have like gill arches, Nilo right here, gill arches, and you have Nilo interconnecting. So that was the first evidence that Nilo is not restricted to the basal teleost, but can also be found in the percomorphs. So if we resume a bit, this is what we have. Basal teleost would have ILT, ALT, and Nilo, while so far the percomorph or modern teleost would only have ALT and Nilo. But still, we have to keep in mind that this remains a very limited overview. It's just a few species. There is more than 30,000, so things are likely to look more different. And sometimes it's also complicated to detect Nilo, especially if we just do section. This is just to give an example. This is a section through the bronchial cavity of Medaka, so done this way. Actin is in yellow, so we like muscle fibers. You have, I think, part of the heart, if I'm not mistaken, or the aorta. You have other muscles. You have the gill arch right here. And there, this is the sternoioidus muscles, if I'm not mistaken, on the pharynx right here. And I will relieve the T cells in magenta now. So you still see this ALT, but it's difficult to say where is Nilo because the organization of the bronchial cavity is a bit different. Is Nilo right here at the connections with the ALT, or is it a bit more at the bottom? So just section is not always enough to determine if we see Nilo or not. So we need to go to more 3D imaging of fish samples. So something we will do with the RC grant. Regarding the amount of samples, so now I also go into research cruise into the North Sea with Shu Wang and Feneric to bring back a lot of different samples. I could bring back more than 30 samples spanning from shark, rayfish to mackerel and galliforms, like a lot of things, and even some weird samples just for fun on art, like baby starfish and squid octopus. I'm not that good as a taxonomist. And of course, I would be interested, if you have samples, you would be interested to see if Nilo is present. I'm interested in collaborations to identify if we find it there, because we have the antibody and we can do imaging with it. So if we resume a bit the bronchial cavity immune system, what we have seen so far We went from a perception that there's only like scattered immune cells on 2 times log to actually have 24 gil lymphoid tissues, 8 ILTs, and 16 amphibronchial lymphoid tissue, to the discovery of a new lymphoid organ that is reminiscent of the tonsils we have in the throat, called NILO. And actually all that being integrated into one superstructure of the immune system by a lymphoid network spanning the entire bronchial cavity. And if we make a summary things to remember from that presentations, yes, the zebrafish can be a good complementary model for more valuable aquaculture fish species, because a lot of the things we found in zebrafish we could corroborate in bigger cyprinids, in carp, and in salmon. I hope from what you've seen that anatomy is very complex in the bronchial cavity, and it's always important to respect the heterogeneity of organ and tissues, because if, for example, if you take the gills, just the gills, if you make a section for the gills, depending on where you look at, you may find a lot of T cells or not, or very few, and that might just be because one of your image is done on the lymphoid T structures or not the other one. So you have to be, it's very important to respect this one. And then I hope you're convinced that the bronchial cavity is an important region of the fish immune system, and I believe that can have a lot, that can be very beneficial for the aquaculture industry. So I would like to thank all the different collaborators. There have been like many, I can't say them all. And yes, for the future, we will study more. All this bronchial cavity immune system is organized on all the different compartments. Actually communicate and exchange, interact with each others through these European fundings. And of course, yes, I am currently looking for a postdoc to start in the 1st of April. So that's it.
Yay!
Yeah, right on time.
Yeah, no, you're perfectly right on time. But I'm applauding, but I hope you can see all the hands popping up on your screen. You get a lot of applause from all the audience. And while I give you some times to, gather your idea, digest the last piece of information you got from Julien. I'll just say, Julien, as always, wow, it's always so impressive. And of course, I hope that this is also inspiring to many PhD students and people that are still in the lab to see how wonderful it really is to sit and stare at your many slides. Takes a lot of effort, but look at the reward that you can get by doing really, really good, outstanding, stunning immunofluorescent microscopy.
And it gives very clear data as well. That's also something that is very comfortable, is that, likely you're not seeing an artifact.
Yeah, it's really, 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 really nice. So the floor is open to questions. Please also open up your camera, show up your face, even if you don't have a question. But please don't be shy. Shoot if you're interested, if you were planning to do something like this. Also ask him, how long does it take? How many hours do you spend? How many hours that postdoc will have to spend behind the microscope if it comes in your lab?
A lot.
Yeah, I can imagine.
No, but
I'm also looking at the chat.
Yeah, we'll have new microscopy. When the project starts, we'll have a light sheet microscope, and we have virtual reality to analyze the images. So everything will be done so that the imaging and analysis is faster and more comfortable.
I recognize Ottavia on the screen. Hi, Ottavia. Nice. Let me see. I see Phil. Hi, Phil. I see Phil has a question. Please go ahead.
We don't hear you.
Try now. Is that better?
Yes. Yeah, we can hear you.
I have a problem with microphones and Teams and things. So, Julien, that was amazing. Thank you. I really enjoyed it. So I was interested in the infection part of your talk. So you showed that there was the rearrangement in the NILO, and it looked like there was a significant depletion as well. So I just wondered, in those fish, were you able to see any connections between the thymus and the big superstructure that you have? And did you see a flow of T cells from the thymus repopulating the NILO, or how do you think the NILO is repopulated with T cells?
That I can't answer yet. For this part, all everything gets replenished. We know that we have cell proliferation. I suspect that a lot, especially from the guillain-free tissue, what we see at the same time as we have this depletion of ILT and ALT, we do observe a lot more T cells and NK cells in the bloodstream and patrolling like in the tissues as well. So I wonder sometimes if they just like there's a disease, they just like leave those guillain-free tissue. It's a speculation, but I suspect that they may just like leave the guillain-free tissue, patrol, and at one point just come back. So which would be very interesting for like studying the homing, just like the circulation of immune cells. Well, exactly the timers, the contribution of the timers to the size of NILO is something we should try to see studying the ontogeny of NILO because it appears very late in development. So this is something that we are doing at the moment.
Thank you.
And actually, while I wait for other hands to come up, are you aware of anybody who has already made a ZEP70 transgenic line or LCK transgenic line in zebrafish, which is photoconvertible, which may also help in doing some of those studies?
Very good. ZEP70, I don't know if there's a line for ZEP70. I think some people try. I know there's a mutant line, I think, for ZEP70.
Yeah. Maybe the LCK one in this case could be equivalent.
I'm a bit. I have some concerns using LCK line because it's something we would have to dig a bit more, take the time to really study more. But I feel sometimes that in at least the old LCK line, it feels that some of the lymph structures does not look exactly the same. If I do the ZEP70 labeling on wild-type fish on LCK, I don't necessarily observe the same thing. So it's something I have to take the time to dig a bit more to see that. I know there's been other LCK line that I used, seems to not have that issue, but again, it's something I would need a lot of time to really
so one
process.
message is be aware which LCK line you have and otherwise complement with the ZEP70 staining. That could already be a message. Okay, now good to know.
But a new line for like photoconvertible would be really amazing.
Yeah, I think that's
I need more postdoc.
Yeah, and another question that comes up at least to my mind, I mean, the CD4 line is already available. So I don't know if you have already started looking at the composition a little bit, because then if it's the relative composition of CD8, CD4, and some of those CD4 can also be gamma delta T cells, not only alpha beta T cells, from what we know, at least in carp. So any
we have
idea?
some preliminary data on that. We had access to CD4-1 line, so we see very well the CD4 macrophages, CD4 T cells as well. We had also access to CD8 line and other lines for subtypes. So we know a bit more what is inside, but I think I still have sometimes some issue to trust some transgenic lines in adult fish, because I'm always afraid that maybe they don't show everything. I know that the CD8 line that we use, if we look into the timers, early timocytes should express CD4 and CD8 altogether, but you see a lot of cells positive for the CD4 transgenes, but you see very, very few cells expressing the CD8 transgene. So this is why I think I would try it with Martin, which is I think the audience as well. Try to go with more single cell RNA sequencing and spatial transcriptomic to properly characterize the different subpopulations.
Yeah, no, definitely true. Yeah, and I think I'm not sure whether Kimball is still connected. Yeah, I see him connected, but I was wondering whether you were going to ask the question: would the NILO ALT ILT structure still look the same in an older zebrafish, where actually maybe the thymus has already disappeared or is about to disappear? And then would this be a structure that would shrink as well, or would be actually the remaining main source of T cells in the body of a fish?
Yeah, so that's a whole project, and I can't answer that for now. We know that NILO is present in all the fish as well, and we have very old fish right now in stock to analyze. But we are waiting now to have the light sheet microscope and do all this 3D labeling to be sure that we have like adequate quantifications of all the morphometrics of different tissues. Because right now, if we do the same thing with Maeva, like each fish that we have to recompose Nilo is just like a whole week doing more than 8 hours a day, just going through sections and recomposing everything. So we need a more efficient way of doing it.
So you're preparing the work for the postdoc. I see Brian. Hi, Brian. Also another board member of the ISDCI from Canada. So thank you. I think it's a bit late for you.
It's 9 o'clock in the morning. I had to get up at 6 a.m. for another meeting, so I'm already bagged. No, my question is kind of related to what Maria asked, but it's like differences between salmonids and zebrafish. Have you looked at what happens in like sexually mature salmonids? Because like, you know, when they're swimming up the river, they've shut off their immune responses. They can be missing 80% of their skin and they don't care. I just wondered if, you know, say in a mature zebrafish versus a mature salmonid, if there's a difference in the regression or the disappearance of this tissue, or if it disappears at all.
Well, it's a good question. I don't know. It's possible. I mean, say like so far it's so new that everything is to be characterized on it, or which react to also toxic like pollutant in the water and that. But it would be interesting. Like I would suspect if they shut down everything, likely for them, if they're about to die because they just spawn, like I think proper adaptive immune system might not be as relevant for them.
Yeah. So I mean, as I said, they're missing 80% of their skin. It doesn't make any sense to have immune responses when your barriers are gone. But it's a good question. Interestingly,
what I can say regarding the crucian carp, that I showed the pictures a bit whoop, there. Where is it? Right here. So the crucian carp, those ones, actually the group that study this crucian carp are very interested in this fish because in winter, where they get them, the lake is fully frozen in Norway, and they survive in anoxia. And they study like anoxia and oxygenation, and they produce ethanol through the gills, or they secrete ethanol through the gills. And I would suspect that Nilo would be quite damaged, or that the adaptive immunity would be shut down and would be a lot less T cells because that's a lot of energy. But still this is like every year it's still back. Like this is after the winter. And you see Nilo very big. So I don't know exactly the dynamic.
When I was back in Wageningen, I did a study with Renee and with Pedro Rodriguez, where we showed that at carp at below 6 degrees turn off MHC class 1. They turn off the beta-2 microglobulin gene, and then there's no class 1 on their cell surface. But if you bring them back up to 12 degrees, it comes back out. So I think that's kind of in line with this. In the winter, it was the same idea. They're not eating, they're not doing much at low temperatures. They're like estivating, so they don't want to waste energy on immune responses. So if they're shutting down class 1, it may be related to these tissues regressing and appearing again, right? So.
I have to say, I would like to have like one fish like caught exactly in winter when they're pure anoxia, and to see if this is like just there or if it's just like gone. We have some infections that we have seen that we know some bacterial infections can wipe out Nilo. Or wipe out also the gill-free tissue and they regenerate. Well, fish is very good at regenerating, so I don't know if this is special of that tissue or just because usual regeneration, but they can regenerate.
Yeah. Cool.
And were you aware of this other organ? Julien, if you go back one slide, I think, where you had the image of the zebrafish, where you could see your entire zebrafish, where you made your summary. There was this other organ that was recently discovered. I think Kimball was also collaborating. It's behind the fin, behind the pectoral fin.
Yes, I've seen one of the presentations from. Sorry.
If you go back to your zebrafish with the fin behind the pectoral fin, so it's good for the PhD students to be aware. Other organs have been discovered.
Yeah, I'm waiting to see for the publication. At the moment, from what I've seen from the presentation, I consider it more as a local unit of the skin on some personal image that I have. But I'm waiting to see exactly how the publications get out.
Well, there's a lot of antigen-presenting cells in fins. Like we have a study I just submitted where if you cut the fins and put them in culture, antigen-presenting cells crawl out like crazy. There's a lot of they're again a tissue that's only one cell layer away from the environment, so it makes sense to have a lot of responding cells in there. So it may be related to that.
Yeah, and there's this other paper. Where is it? 2023 of all the T cell kind of all around the skin.
The zebrafish. That is very interesting. I think the skin has a very specific organization. Also, something we see in the section that actually this tessellated lymphoid network is also within the epidermis. So most of these big aggregates are still within the epidermis of the skin, not beneath. So it's very interesting, like all the dynamic, all the cells like move around the body. And if we see that in zebrafish, which is rather small, would we see that in bigger fish where maybe the distances would make that a bit less efficient? No, it's very cool stuff in zebrafish so far.
Yeah, and this I hope for some of the young people, either whether you are a zebrafish lover or whether you are, the rest of the fish lover, you really see that there is a lot of, I always say Julien is a very good example of how you can learn from all these fields that sometimes feel a little bit separated, but well, at least we have the experience that we can combine them very, very well. So if you can try to, take the best of all the models and all the worlds that you are working with, and most importantly, reach out to people that indeed work on different models, because there's so much to learn from one another, even just for the tools. As you have seen, one antibody that can cross-react on so many species. I mean, that is such a valuable tool, right? And if you cannot trust if you want to complement what your transgenic line says, then reach out, for other tools and the expert that use them.
Yeah, I would really recommend, if you can, sometimes just not dissect just your organ of interest, but look at the overall picture. See, like your transgenic line, you see the signal there, but you should also look, okay, I see signal here that should not be there, and vice versa. I think we need to have a bit more characterization from the transgenic line in adult. Some things that we think are weird might just be true biological phenomenon, but some might be some leakage or artifacts. Like, we have to be careful with this.
Yeah. True words. Now, I think time is running out. You can also check the chat afterwards, Julien. You got lots of compliments from some people had to run out, from Natalie, Louisa, from Phil, from Kimball. So you can read afterwards. I think everybody really, really enjoyed it. I've seen your presentation a couple of times and I never, ever gonna get tired of it. So I think I'm gonna invite you again. I'll find another excuse. It's really, really wonderful. Hope it has been inspiring for many of you. And last but not least, if you have any idea, suggestions, or you have a favorite speaker that you would like to see featured in one of our next seminars, we're composing the program for next year, so we'll resume somewhere next year in February with the seminar. Please let us know. You can send me directly an email, or you can send an email to the ISDCI email. You can check our website, and also I can announce it here, and some of you will also receive news letters through your PIs. The ISDCI has also launched the possibility to have travel fellowships. So if you are a member or your PI is a member of the ISDCI, as of next year, we give out fellowship to attend specific congresses that are in the field of comparative immunology, or you can also travel to another lab to have a training period in another lab. So check out our website for the conditions, and of course ask, or you can apply, or your PI can apply. Okay, this was all from me. Thank you all for joining. Thank you, Julien, for your wonderful presentation, and we'll be in touch through the newsletters.
Yep.